Okay, this is the structure of mitochondria. So we need to remember it has two membranes, outer membrane, inner membrane, and the foldings are called cristae, and the inside space is called matrix, and the space is called intermembrane space. And the 6 and 7 features are 7th ribosome and circular DNA. At least 7 components you should be able to draw in your picture. And this is the electro micrograph, how it looks like. There are two functions. Function number 1, production of energy. That is the ATP molecule. Function number 2, photorespiration. So I again tell you a photorespiration. We're going to learn it. So I just have a space. I quickly show it again. So this is the chloroplast. During photosynthesis, at special event, it produces a molecule, and the molecule is first going to the peroxisome, and then this molecule is moving to the new molecule, right? That's a different molecule. It changed. This is converted to different molecule, and that is going to the mitochondria. And this molecule again changed to different one. It's going back to the chloroplast. We need to learn in photosynthesis, but peroxisome and mitochondria take part of doing this photorespiration. So mitochondria has two functions: production of energy and photorespiration. Okay. Okay, I think it's enough for the mitochondria, and only thing is we want to find why mitochondria has 70th ribosome and circular DNA. It's like this. If we, and the next prokaryotic and eukaryotic organism, we're going to learn somewhere around 3.5 billion years ago. The prokaryotic cells are formed. The first cell is formed, believed, in 3.5 billion years ago. That's a life established on Earth, born of this prokaryotic organisms like a bacteria. Then after that, somewhere around 1.8 billion years ago, BYA, the eukaryotic cell formed. So then eukaryotic cells, as we learned, they have an 80th ribosome. And this has 70th ribosome. And it has own DNA. And this DNA is here. That's basically the eukaryotic cells and prokaryotic cells. Then scientists are believing now, some point during this formation, these prokaryotes move into the eukaryotic cells and move, and also change to become mitochondria. And same times chloroplast in plants, and this in animals. So this is the explanation given. The earliest cell form is prokaryotic cells. Later on, eukaryotic cell form, and the prokaryotic cell move into the eukaryotic cells, and it went inside, and it changed its shape, slightly change, and start to live permanently. And they say this is the theory again, because you learned cell theory, something based on some evidence. Scientists proposed a concept that is a cell theory. Now, this is a second theory. This is called endosymbiotic theory. Now, what is endosymbiotic theory? Endo means inside, living inside. Symbiotic is the relationship between two different organisms. So that's a relationship between two different organisms, the prokaryotes and eukaryotes. So the benefit is this produces energy as ATP, and from this cell it gives nutrition like glucose. Eukaryotic cell give glucose, and mitochondria give ATP. So then benefits each other, and this is called endosymbiotic theory. That's the explanation given to explain why that cell has 7 ribosome and circular DNA. I think if you copy this one to the notes, it's enough, because all what we have the text again in the resource book. I mean, we don't want to write, and if you want, you can download the different version of the one written as numbers and highlighted important things. So chloroplast, mitochondria, has 2 functions: production of energy, ATP molecules. Second function is photorespiration. Photorespiration, we're going to learn in photosynthesis. And also we give the answer for why they have 70th ribosome, explaining this endosymbiotic theory. So prokaryotes born 3.5 billion years ago, after that eukaryotic forms, so they merge, they fused to become one cell, and that kind of relationship is called endosymbiotic theory. So this is the structure of mitochondria with the Electro Micrograph. Next, similar structure to mitochondria, chloroplast. This is also very similar certain aspect in mitochondria, because they have 2 membranes. Outer membrane, and the inner membrane is not folded. Now, mitochondria, you have seen a cristae, the foldings. But similar thing, we can find it here, but they are not the not exactly how it folds this inner membrane, but there's slightly separate disc formed. Like this. Many discs increase the surface area inside. And the disc is called, one disc is called thylakoid. And collection of this thylakoid is called granum. Plural is grana. The plural word is grana. And this one granum is connected with other granum like this. This structure is called lamella. And this one is called not matrix, this time it's a stroma. In mitochondria, it is matrix, here it's a stroma. Interestingly, we can find a ribosome with 70S and circular DNA. Circular DNA molecules. So this again prove the concept of endosymbiotic theory. So the photosynthetic prokaryotes move into the eukaryotic cells and it becomes a plant cell. We can also find a starch grain. The starch collection of starch make a big structure, starch grain. Again, if you can label like this and draw the structure, that's enough to remember all these things. The main function is photosynthesis. Chloroplast. Here it looks like in a 3D picture. You can see inner membrane and the outer membrane. And then you can see some kind of a disc like this. And this disc are connected with each other by lamellae. Here's the lamellae. I don't know whether you can see the cursor. This is the lamellae. Yeah. Okay. So we started here today, Golgi complex. And also we learned the protein transport. And after that, we learned the 3 functions of Golgi. And lysosome also we learn. And lysosome again, we learn 3 and 4 different functions. 4 different functions. Digestion of food particles, phagocytosis. The breakdown material inside the cells. Digestion things inside the cells. And transport the unwanted thing out of the cells. And also the digest organelles which are not really used up. And also autolysis, that means cell death. It's again action of lysosome. Glyoxysome, the main function is detoxification of hydrogen peroxide. peroxisome detoxification of hydrogen peroxide, and also photorespiration. Glycosome plant's version, so it's mainly lipid convert into a carbohydrate sugars. So mitochondria has two functions, production of ATP and photorespiration. And chloroplast has photosynthesis. We're going to learn something very different structure called cytoskeleton. The cyto refers to the cell, and as usual, the skeleton means just skeleton like we have, the bones. So our body is made out of bones, that's called skeleton. Then cell also has a skeleton like that. But surprisingly, we haven't seen all these pictures, the cytoskeleton. When you look a lot of pictures, we haven't seen such thing, but it has cytoskeleton. If you want to see cytoskeleton, we want to see a different way of the cells. That means we have to color the cells with different colors. Then only we can see the cytoskeleton. Right, I think I can show the cytoskeleton in a picture.
If you take a look at most.
Okay, here you go. Now you see a lot of structure that you learn: mitochondria, nucleus, nucleolus, RER, SCR, vesicles, Golgi. You see some kind of cables like things distributed all over the cytoplasm is actually the cytoskeleton. So here, if you look, this one, look at this picture, like this kind of a pictures. You can see inside so much of fibers like things. So they represent the cytoskeleton. That means many cells, they have this inside cytoskeleton. So if the cell is like this, there's an extensive network of things, fibers like things distributed inside the cell. It's a cytoskeleton. Here, another picture. You can see the cytoskeleton after staining with different staining called fluorescent staining. You can see a very extensive fibers distributed all over the cytoplasm. So it's very close to the cell membrane, and it's even very close to the nucleus. From nucleus to the cell membrane, this network is extended. That is the cytoplasm, the cytoskeleton. Okay, we will come to the detail of these fibers later on. The one is providing mechanical strength, movement of the cells, so chromosome separation in mitosis and meiosis. We will see a third one later on, but you can understand it provides the strength to the cells because cytoplasm and the cell membrane is the fluidic. Cytoplasm is fluid-filled sac. It gives the strength to the cells, and the fibers can move around, and they can provide the movement of the cell's locomotion. Third function, we're going to learn mitosis and meiosis, and we can learn that, right? And then the cytoskeleton is made out of protein filaments. One is actin filaments, also called microfilaments, and the other one is called intermediate filaments. And the third one is called microtubules. So three kind of filaments: actin, intermediate, and microtubules. They make the cytoskeleton. If we look what is the structure of microtubules, you can see there are 13 protein molecules sit to make columns. Like this: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So these are the protein molecules. And then they extend like this to make the tubes. So inside is empty, and it consists of these 13 protein columns. So that's another protein, another protein like this. They join together to make this column. And the name of one protein is called tubulin. So this name of the protein is tubulin. Here you see it again, the same structure. So hollow, the lumen, that's the empty middle, it's called lumen. And the wall is made out of tubulin proteins. So they organize as columns. Now we're going to look separately function of these three filaments. So here we generally learn the three functions of cytoskeleton. Right? The cytoskeleton is made by the three filaments, right? The cytoskeleton is made by three filaments: actin, and the intermediate filaments. Actin filaments, microtubulin, and then intermediate filament. Actin is also called microfilaments. So three filaments made this cytoskeleton. 1, 2, 3. The general function of cytoskeleton is strength, movement, and important in cell divisions. So microtubules is the first one. So microtubules made out of proteins called tubulin. So 13 protein molecules make the columns like this, and they make these long filaments. So we have to always remember all these three filaments. If we take as a single filament, they all are important for the strength of the cell. So this is important for the strength of the cell. One function of all the three filaments is giving the strength to the cell. We can say it's a cell support or strength, or support or strength to the cell. Second function, a chromosome separation. In cell division. This we're going to learn in detail in mitosis and meiosis. So what is chromosomes? Even the tinier details of chromosomes and how it attached to these microtubules and how it separate from the microtubules. So we're going to learn in detail this function. Third one, within the cells, movement of organelles. Movement of organelles. These are the three functions of the microtubules. And another important thing we're going to learn, that is this one, Syrian Flagella. We're going to learn Syrian Flagella. So it's basically the microtubules. I think we can add the fourth one. Syrian Flagella. Strengthen the cell, chromosome separation, helps to move organelle within the cells. It helps to make this Syrian Flagella. So these are the four functions you need to remember, and out of the four functions, this one is always common to all the three filaments. Strength, providing the strength to the cell, is common for all. So then the 2, 3, and 4 is important to remember for microfilaments. Okay, the next one is actin filaments. So actin filaments again make the protein called actin. So one is running like this. The other one is also running like this. The joint intertwine together to make this actin filament. And if I want to show actin protein, this is actin protein. So actin filament function again, strength to the cell. Second one, it make the muscle contraction, important in muscle contraction. Number 3, this one, when we're going to learn mitosis and meiosis, that means nuclear divide to make 2 cells. So that's a common cell division. So initially it's like this. This side has 1 nucleus, the other side has another nucleus. And in the middle somewhere here, a large amount of Golgi and also RER, they're going to organize in the middle to form Golgi and the RER. After that, the cell is trying to break from the middle like this. And this, the breaking points into the middle is called cleavage furrow. So actin is important to make the cleavage furrow. Actin form the cleavage furrow. This one again, we're going to learn in cell division. The 4th one, can you remember here, the one of the function of lysosome is phagocytosis, help in phagocytosis. Now I said the cytoplasm makes some extension. So I did not use any name for that extension. These are called pseudopodia. These are the pseudopodia, cytoplasmic extension. And here actin is important. Actin is important to make the cytoplasmic extension. Because cell membrane has to move, but actin filament push them to make the pseudopodia. I can share this file at the end if you miss something. I will write here again the function of actin. Pseudo is not correct, not true, like fake. Podia is an arm. So it's like a fake arms. Pseudopodia, the meaning is a fake arms. So there is again 3 function important. Strength is anyway common to all these fibers. Muscle contraction, actin is important. Cell division to make the cleavage furrow, the middle breaks. And it produces pseudopodia, example in phagocytosis. So that is again 3 major functions we can remember under the actin filaments or in microfilaments. Okay, today we can finish the last thing. Intermediate filaments. That's the last one. So they are formed a collection of proteins. It's not a actin make two filaments, and then microtubules make 13 columns filament. And here we don't have exact number. It could be like 10, 12, 13, 14 around the protein filaments like keratin. Just unfortunately the pen stuck, it doesn't work. All these technical problems. Yes, I think we have a small issue. I can't write here now. The program made some problems, so it doesn't work now. Okay, anyway, it's end of the time, like it's a 1 minute left. I think what I can do, I can stop the class today. If you want this document, I can share. If you miss something, you can add to your note section. And I think the important is the understanding. Right. So I'm going to stop here and we meet again next time, right? Okay. See you
Thank
again.
you, sir.
Okay, bye.
Thank you, sir.
See you.
Thank you.